Our first speaker today is Natasha Caplen from the genetics branch. She got her Ph.D. from the University of London, King's College Hospital Medical School. And subsequently, she did postdoctoral training at St. Mary's Hospital Medical School, Imperial College. And in 1996, she joined the National Human Genome Research Institute at NIH as a visiting fellow. She started working on siRNA. And she's now a investigator in the Center for Cancer Research, and she's going to talk to us today about functional genomics. Natasha.
All right. Good evening. I know I'm okay in here. Am I okay across the land? Oh, they're not telling me I'm not, so I'm assuming all okay. All right. Okay. So thank you very much, Terry. Thank you for the introduction. So what I'm going to do today is give you a sort of an overview of my take on the functional genomics field as a whole. I'm going to do a relatively brief introduction about some terminology and some key concepts that I just want to get in place. Then I'm going to spend a little bit of time on the technology, but I really want to spend most of the time today talking about applications, predominantly using work from my lab, to give you some concepts about how we can use different functional genomics technologies. I'm going to really focus predominantly on RNAi-based technologies, but really a lot of the approaches are going to be the same, irrespective of how you actually manipulate gene expression. Okay, so why do we need functional genomics? So we know that the technological advances that we can now use to assess things like DNA copy number and sequence, and I'm really here thinking about it in the context of cancer, that we can look at DNA and chromatin modifications, and we can look at gene expression on a very, very large scale and a very great deal of depth. And that has revolutionized how we understand the molecular disequilibria that is underlying cancer and obviously many other diseases. But our ability to functionalize these findings in a very systematic manner, it still remains a challenge. And so for the last 10 plus years now, there's been a lot of emphasis on trying to develop functional genomic strategies that we can conduct on a similar scale and then drill down into what these types of changes that we're seeing either in DNA copy numbers, sequence, looking at chromatin modifications or gene expression, what this actually means in the context of looking at normal function and then obviously in the function of genes in a disease setting. So the model systems that you can conduct functional genetics are obviously as broad as all of the different model organisms that we have available to us. On a gene-specific level, you can do a lot, or even on a large scale, you can do a lot of these experimentation in bacteria, yeast, C. elegans, zebrafish, Drosophila, and mouse. In the context of human, most of the work I'm going to talk about today has been done in transformed cancer cell lines. And I'm going to give you a hint of some of the advantages and disadvantages of that as we go through. Increasingly, though, particularly with some of the new technologies, you're seeing it being able to be applied to primary cells, into patient-derived xenografts, into PDXs, and then induced pluripotent stem cells. As I say, what I'm going to talk about predominantly is in cell lines, but it can be extrapolated across. Okay. So there are really sort of two flavors of functional genetic analysis that I'm going to talk about. I'm going to predominantly talk about the second of these, but I just wanted to mention this first, which is the gain of function. So it's when you're overexpressing or you're sort of replacing a gene to see what its effect is in a different context. So you can overexpress a wild type gene. I want to make sure I'm getting the, yeah, it's not very. You can overexpress a wild type protein in a mutant background to see what the functional effects are of that. You can use overexpression in epistasis tests where you can overexpress different genes and look at how that affects a particular phenotype. Where there's actually probably been most work is in the overexpression of a gene, that usually the open reading frame, the cDNA, in a heterologous system, either a whole organism or in the cell lines that I described earlier. And these can be used to look at wild type proteins or mutant proteins. They can be used to create interaction networks, and they can be used as modifiers for screens or looking at drug targets. So there's really a very large way and different ways you can use these types of overexpression studies, but there has been a tendency to actually not do this on a large scale. This is hard to manipulate, and it's been hard to read the phenotypes from these. So there's been most emphasis in the functional genomics field really on a loss of function, where there tends to be a clearer phenotype at the end of it. So there are two types of loss of function in the crudest type way that I'm going to look at. You can have a knockdown, this is really not, or you can have a knockout. Okay, so I'm going to talk about both of these, but they are different. Okay, so when you knock down, you've often still got a residual level of protein expression, or certainly an mRNA expression there, and that can you really need to think about that in the context of the phenotypes that you're investigating. A knockout, in theory, should take out all of the protein or all of the RNA molecules that you're targeting. So you have a difference that needs to be borne in out. So the other piece of this puzzle that I just want to put forward is the difference between forward genetics and reverse genetics. And then I'm going to pull all of these together to discuss the different technologies that are available to us. So forward genetics is the mapping of a phenotype to a mutation with no prior genetic information. And for a long time, this was done in the context of mutagenesis studies using chemical radiation or viral mutagens. The other is to do reverse genetics, where you're using prior genetic information to generate the reagents that induce a phenotype. So for a long time, and particularly in mammalian systems, we needed to do forward genetics because we didn't have all of the information we needed to do reverse genetics, which for a long time was really the purview of things like yeast or Drosophila models. But obviously with the identification, the full sequencing of the human genome, and then with the tools that I'm going to talk about today that we've developed, increasingly we're now able to do reverse genetic studies. So I'm now going to sort of move on to the technologies in the context now just of loss of function, okay? So they really come in two types. Those where you're focusing on gene silencing, which is often, as I say, is referred to in the literature as sort of a knockdown, and is predominantly using the RNA interference mechanism. The alternative, which we've now seen increasingly now become applicable, is to do gene editing, where actually this can be both either a knockdown or a knockout, depending on the effector molecules that you're using. And these come in two predominant flavors in terms of technologies that have now developed over the last few years. One's using transcription activator like effector nucleases or TALENs, and then the ones that obviously use clustered regular interspaced short palindromic repeats, or CRISPR. And I'm going to go through these in a little bit of detail just to give you some basics. Now, there has been a plethora of different reviews over the last few years that have been looking at these different technologies. I'm going to focus on one that came out actually now a couple of years ago, but really is still relatively, how am I going to say this? It's very relevant still. A lot of the issues haven't changed that I really want you to focus on. And the key thing here is, and why they've titled it was choosing the right tool for the job. And it's the context is that actually what is really great now is we have options. We can try all of these different things, and we can see which is going to work in the context of a particular experiment. So I'm going to walk through these three different main areas. I'm going to talk about RNAi very briefly, then go on to TALEN, then go on to CRISPR-based techniques. So RNAi is a post-transcriptional mechanism that induces RNA degradation. And so this is a reversible knockdown. And the major ways of inducing the effect is to use a small interfering RNAs. So the RNA mechanism is a naturally occurring gene silencing mechanism. It's predominantly known in the context of using microRNAs that regulate post-transcriptional gene silencing by interfering or interacting with the 3' sequences of a transcript, and that alters the expression of the protein from a particular transcript. But we've been able to exploit this by using SIs or shRNAs that actually target specific transcripts and induce a degradation of that transcript. Now, one of the issues has been that there is quite a large off-target space for this. Effectively, there are, because of the way that microRNAs work, you can have an interaction with an siRNA with a lot of different sequences, and so that has been one of the major problems with this. I'll mention how this can be overcome in a few minutes. But it's still very robust, and it's very quick in comparison with CRISPR. You can do an siRNA experiment in a couple of days, where still doing CRISPR experiments is going to take you a long time. So sometimes a lot of the question is simply, what is the question I want to know, and how quickly do I want to know it, as to why it would make sense to use an RNAi-based experience. So just to cover what I just mentioned in terms of how this works. So predominantly, we're using siRNAs or shRNAs. So siRNAs are synthetic molecules that can now be purchased from many different commercial sources using fairly robust algorithms. Typically, one will test at least 3 to 4 different siRNAs per gene to be sure of successful knockdown. shRNAs are going to be expressed from a plasmid, and they can be expressed also from an expression vector. This is targeting, then I said, the RNAi pathway. I'm showing a highly simplified version of this, where I'm just focusing on the cytoplasmic part of the pathway, where it's going to interact with the RNA-induced silencing complex, predominantly with the protein AGO2, which will take one strand, it will take the guide strand from the siRNA or from the shRNA that's been processed by DICER. It will align that on a sequence-based, and it will cleave in the middle of this. This will reduce protein synthesis as a consequence of the fact that you have degradation of the transcript, and therefore then a loss of function. So the assays that can be conducted downstream of this are fairly obvious. They're going to be mRNA, protein, or function. And what really has to be optimized to apply this technology is making sure you've chosen the correct model system to be able to look at your particular question related to loss of function, the efficacy of the particular reagents that you're using, and then, as I mentioned, specificity. Now, the talents are sort of an in-between stage, if you like, between RNAi and CRISPR. And what they're using is a way of inducing a frame shift DNA mutation, or as I'll talk about in a different way, you can actually repress transcription, which sort of becomes a little bit more like RNAi. When you're inducing the DNA mutation, obviously this is a permanent knockout, whereas if you're using an effector molecule that induces a repression of transcription, this can be reversible. So the required information for this is the transcriptome, which is the same as with RNAi. But what's key for tail repression is you actually need to know exactly where the transcriptional start site is for your gene. And that's not always very clear, and it's not always very amenable. So the off-target space for this is thought to be a little, particularly for gene editing, is thought to be a little less, because as you'll see, you need a lot of sequence to define where you're going to edit, and that should reduce the off-target space. But you can also, in the context of repression, you can see that that may not be as specific as you would like. When I talk, I should have stressed this here in a minute ago. So many genes have transcript variants, and that's one of the things you really have to consider when you're designing these experiments, that if you're going to be editing a gene or silencing a gene, are you going to want to take out all of the variants, one subset of variants, because these variants may have different functions, and so that might be come into your thinking. All right, so I'm going to talk a little bit more about tails or transcriptional activator-like effectors that can be used for programmable DNA binding proteins. So these are talents, and they use synthetic proteins that are based on the protein secreted by the Xanthomyrmex, which is a gram-negative bacteria that can infect a wide variety of plant species. Okay, and so these have DNA binding domains, and that's what's absolutely key about these. So they contain 7 to 34 homologous direct repeats of these 33 to 35 amino acids, and these can recognize a specific DNA motif that is also still present in mammalians. So the specificity of each of these tails, each of these repeat sequences contained within the 2 amino acid positions that are at 12 and 13 of each repeat. I'm going to show you a picture in a minute that will make more of this clear. And these are these repeat variable diresidues. So that's where the specificity comes in. Now, the key thing is each target site for these tails in the genome requires a different DNA binding domain of 500 to 7,000 amino acids. So this has made it not as flexible as many of the other technologies available because you require to build it like this. And so here is that model that I was showing, and you need an awful lot of space, of DNA space, to be able to build these different domains and find exactly the sequence that you're going to be able to cut with this. So the nuclease effectors are these FOC1 domain that acting as a dimer to introduce the cut, the DNA double-stranded break. And so you actually should have minimal off-target effects because you've got to have both of these sequences aligned correctly to get a cut. But the problem is that you have to design each of these, and these are very expensive. So this is not being as widely used, but it does have a lot of specificity. So the way it will work then is through either DNA repair, through homologous recombination repair, where if you include a donor, or you can integrate in a sequence, or it will introduce non-random point mutations, targeted deletions, or as I say, if you put in a donor, you can get in a DNA fragment. Equally, you can have error-prone non-homologous end joining, and this will induce deletions, insertions, or substitutions, and that's where you're going to get your loss of function phenotype, because it's going to have a frameshift mutation or it'll truncate your protein. Now, the way it has been adapted by several groups to try and not induce repression rather than a complete DNA editing is to use a Crupple-associated box, a CRAB domain, which is then fused to this, one of the DNA binding domains in this case, that will target a transcriptional start site. So effectively, it will just sort of sit there and it will block the ability of the transcription to occur at that site. Now, the disadvantage is you have to often test different sites within the transcriptional start site to avoid the occupancy of where the core promoter is. Because you've got to be able to actually interact at the site to block where the transcription factors are going to be bind, or where there's high nucleosome. So actually being able to design these can be quite difficult. As a monomer, these can induce off-target effects. That was the advantage of here, where you have the double. This is less non-specific than here, where you tend to have a risk of off-target effects, because trying to find these sequences can be quite difficult. But a lot of groups have used them. They've been less used in the context of screening. So now I want to turn to what I'm sure everybody in this room has now heard of, and actually just out of curiosity, so how many people have actually done CRISPR experiments? Just Nick. Okay. I'm trying to get a sense, across the campus, we're trying to get a sense actually of how many people, so how many people have done sRNA experiments or RNAi experiments? Okay, all right. And how many people have done TALEN experiments? No one. Okay, all right. And I know I can't see what everyone looking on the web is doing, but we're just trying to get a sense across campus how much people are using these types of technologies at the moment. So obviously you can't at the moment open your newspaper and not find any, somebody talking about what people are doing with CRISPR at the moment. I'm not going to go into, I could do a 2-hour lecture on CRISPR. I am not going to do that. So I'm just going to give you some basics just to make sure everybody's up to speed. So there really comes in 3 major flavors. So this is, as I say, changing by the day at the moment. One where you're predominantly using a highly active Cas9 nuclease that will induce a frameshift DNA mutation and that is therefore a permanent knockout. So this is more like the, you're making a single, you're going to make a change that is going to completely knock out a particular gene. If you've designed the targeting the guide molecule, which is a guide RNA, correctly. So this information you're going to need, ideally you're going to need to the whole transcriptome, that you're actually going to need to the genomic information too, because it's going to cut the genome. Okay, there is an off-target space across the genome, and I'm going to talk about that in a few minutes in this context. And you have the potential to be able to target loss of variants if you know exactly where those are all mapping to. Now, there are now increasingly a couple of other flavors of CRISPR where you are trying to do repression of transcription or activation of transcription in a way that's very analogous to, in the case of to RNAi, or where you're using what I've just shown for the talons, where you're again putting on a crab domain to block expression at a transcriptional start site. So this is really the same as I just described for talons. And then CRISPR-A is where you're activating, where you're using a VP64 to actually activate expression. And I'll show a picture for that in a minute. Okay, so Cas9 is the CRISPR-associated protein 9 nuclease that was identified in the Streptococcus pyogenes and this is the major source of Cas9. There are now increasingly lots of other Cas molecules that are being found in different bacteria that have slightly different properties. Most recent one of which is Cas13 that appears to be able to act to edit RNA. So Cas9 has innate nuclease activity, and the targeting is achieved using the CRISPR RNA, which is shown here, and a transactivating RNA, which can be formed as a single guide RNA here. Okay, and I'm going to go into this in a tiny bit more detail. All right, so the constant region of any guide RNA is going to interact with the Cas9, and then the variable region is going to interact with your genome target of about 17 to 20 nucleotides. And the way that that is recognized is by what's known as the PAM sequence. So this is the protospacer adjacent motif, or PAM, that is a short region immediately to 3' of the sequence that you want to target. So here's it represented here, okay? So you need to know exactly where the PAM sequence is, and then you can predict where the Cas9 will cut. So the PAM sequence for Cas9 from Streptococcus pyogenes is NGG. So as you can imagine, there's a lot of those. Having said that, there are still some parts of the genome where actually there isn't one that's convenient. And so in those cases, actually what individuals are doing is actually going back to talons, because you sort of, as I say, it's about having the right option for your particular question. So here is showing the different types of gene editing strategies that exist. So like I just said, for talons, you can have non-homologous end joining, which will result in an insertion or a deletion, which if it induces an out-of-frame mutation will mean that now this gene will no longer be expressed. If you add in a donor DNA, you can then get homology-directed repair, which you can use to insert a gene. And this can be very useful as a way of tagging genes, either at the 3' end or the 5' end, to maybe put on an epitope tag or to put on a fluorescent tag that you can use for imaging. And this is now increasingly being looked at on a larger scale. So you can also, and this is becoming very important, is you can actually now get a much more precise homology-directed repair by actually using 2 different molecules to allow you to insert. And so one of the approaches that this is being used for is even to try and model translocations. So you're actually trying to, if people are trying to take translocations that have been seen in different tumors, they're using multiple recognition sites to actually try and induce that translocation in model systems. So I mentioned a minute ago that we can also use a nuclease deficient Cas9, so these are known as dead Cas9s or dCas9s, that you can then put on with effector molecules to also manipulate gene expression. So in the same way as I mentioned for talons earlier, you can put in a crab domain that will act to repress gene expression if you can target exactly at the transcription start site. This is done typically about 500 base pairs downstream for the same reason that, again, you need to find a low occupancy site for the molecule, for the Cas9 to interact with. For gene expression activation, usually it's fused to a transcriptional activator such as VP64, which will then actually induce expression. And finally, one of the other options that people have been using these types of technology is actually you can actually add on a fluorescent protein just to the Cas9, and you can use that for visualization purposes. And so that can be has been used by a lot of groups. So I just want to stress about the off-target effects on this, because this is going to be relevant as we go through this. So in the context of Cas9, or in the context of CRISPR, there's a couple of pieces of information. So the proximal 8 to 14 nucleotides that are immediately upstream of the PAM are the most important for target site cleavage. And so you've really got to look at a lot of different options when trying to design where you're going to be targeting. And in this case, you can have the Cas9 combined to a large number of off-target sites, and the levels of the guide RNA may also be contributing to the off-target effects. And I'm going to talk about how groups have been looking at addressing this in a moment. So one of the ways to do this is by making mutations in 2 Cas9 nuclease domains and turn the Cas9 from a nuclease into a nickase, that then you can use the 2 to target adjacent sites. And so this is one attempt that groups have been making to try and get around this problem. So there are lots and lots of different options for trying to make guide selection. And there are increasingly commercial sources, but almost all of those will tell you to test at least 3 or more guide sequences, just in the same way as I said you need to test multiple siRNAs. Because here, it's not just about specificity, it's about efficacy as well in both of those cases. But I want to stress I am not advocating for either of these algorithms. I just pulled a couple that are currently available that you might want to look at for doing guide selection. One is from George Church's lab at Harvard, and the other is from Greg Hannon's lab, who's now in Cambridge in the UK. So these allow you to try and predict what might be the best guide RNAs to target your particular gene of interest. And one of the things that's a feature actually of Greg Hannon's is that they've really focused on these dual guide systems. So they have 2 guides to try and improve this specificity. Now, one of the other features to consider is where you're going to target within a gene using these guides. And I sort of just wanted to point out here, and I apologize, I realized that this figure wasn't in the printout. But these are 2 studies that came out in the last year. Also, one here from Chris Vojak's group at Cold Spring Harbor, and then this I think was from, I apologize, I can't remember where this one was from. I think it was from the Harvard groups, where they showed in 2 independent studies that actually guide RNAs where you're targeting a specific gene to try and induce a complete knockout phenotype, that if the guides were targeting the functional domain, so what you're seeing here is this is the functional domains for one particular gene, and these are the key domains that we know are involved in the function of this protein, that the guide RNAs that targeted these regions are more likely to induce a loss of function phenotype than those that are targeting domains that are not important to the function of the protein. And you're seeing this here for a second gene that you see in these where you're targeting the ATPase site, you see more effect. And this was actually seen in exactly the same in this study, also looking at the beta-catenin gene, where these proteins that are involved, these particular guide RNAs that were targeting the armadillo repeats within the beta-catenin gene were much more effective in inducing a phenotype than those at the 5' or the 3' end. And you see this again also in, for example, in the urokinase B. So one of the thoughts is that if we induce these mutations in highly functional domains within proteins, that you're going to get a much more robust phenotype. So in the last, part of the half, sort of 20, 25 minutes, what I really want to do now is take some of the things that I've talked about and now apply it in the way that we've done it in my lab, and then throw in a few pieces of information from other people's lab. I said I'm going to focus predominantly on RNAi-based studies, but the types of ways you can use these effector molecules is basically the same in terms of thinking of the downstream work from that. And I'm going to talk about it in the context of where we've gone from a screen, so we're actually a genome-wide scale study, then down to specific gene function analysis, and then I'm going to finish with looking at how we've looked at it, use these types of techniques to look at the interaction of genes and drugs. Okay. So I said earlier that my lab predominantly uses siRNA-mediated RNAi, so we're using small synthetic siRNAs, and what we're in a position to do is actually scale this up through the work of the TransNIH RNAi screening facility that's actually administratively held at NCAT, is we can scale this all the way up to genome-wide scale. And what I'm showing here is where we've actually conducted a screen looking at the activity of a fusion transcription factor, EWS-FLI1. And we've done it by looking at a reporter assay that involved two different cell lines. Same cell line, but they were what we call isogenic. One expressing a reporter that will be a readout for EWS-FLI1 transcriptional activity, because you have a luciferase reporter that is fused to a promoter that is recognized by the EWS-FLI1 transcription factor. And then as a control is a cell line where we have a reporter with a CMV, a constitutively expressed luciferase. And what we were asking was the question, if we silence a gene, so if we silence gene A or we silence gene B, what is the difference between that effect on this reporter assay versus this reporter assay? On the basis that if we saw effects just or predominantly in this cell line, where we are looking at the effect of the EWS on the EWS-FLI1 target gene, would we find genes that were involved in the expression of this fusion protein or the activity of this fusion protein? So why do we care about EWS-FLI1, and why am I even showing this to you? So EWS-FLI1 is involved in a relatively rare but pediatric tumor called Ewing sarcoma. And Ewing sarcoma, what you see is that these typically have a chromosomal translocation involving chromosome 11 and 22 that makes this fusion gene that I mentioned just a moment ago, EWS-FLI1. And so the key thing about Ewing sarcoma is that at the moment we have an overall survival rate of about 60 to 70% in these individuals, but it's associated with very intensive chemotherapy and radiation and surgery in a very young population. And in fact, if there's metastatic disease or they are refractory to this chemotherapy, our overall survival rate is less than 25%. So we're trying to find new ways of targeting this, and this was what the basis of the study that we set out to do using an RNAi screen. So what I'm now summarizing, if you remember, was that reporter assay that I talked about just a moment ago, and I'm now summarizing here about 2 years' work in one graph. Okay, so what I'm showing you here is actually a way where we can look at the entire genome. So this is where we've looked at every single one of these dots represents a gene in the genome, and where we've silenced it with 3 different siRNAs, and we've compared the effect of silencing those in the CMV, that constitutively expressed reporter, versus our experimental, which is the EWS-FLI1 readout. And what I'm just going to point you here is these were the control siRNAs that will take out the fusion gene, and then what we're looking for is anything else that has a similar effect. Genes that are going to affect transcription globally are going to be down this axis here, okay? So what we can do is score the effects of this. So I'm showing here for the controls where we have the siRNAs that we're looking for is the difference between these 2 different screens. As I say, on a genome-wide basis, I'm showing you the controls here. So we're looking for these values that are very negative, in this case a z-score, where we know that these siRNAs are affecting selectively the NR0B1 assay and not the CMV. And this is how we can then rank these. So this is looking at the top genes when we've gone through 21,000 genes. This is where we're now looking at the rankings of all the genes across this genome-wide screen. So I'm just going to walk you through one of the genes that came from this to give you a hint of some of the techniques that we use and how we use siRNAs to then probe down our individual gene function. So here we're looking at H&R and PH1. So we found one of our lead hits shown here. So these are the genes we're really interested in, because here's FLY1, here's EWSR1, and here was one of our hits, H&R and PH1. So here we've now validated this. We've now gone up to using 6 different siRNAs, and we see here that when we silence H&R and PH1, we get effects in this assay that are comparable to if we silence the EWS-FLY1 gene directly. We see that when we actually look at the mRNA levels, and then this is a very typical type of experiment you're going to conduct if you're going to use siRNAs to look at gene expression. If we silence H&R and PH1, so I'm showing QRT-PCR data showing the silencing of H&R and PH1, we see that EWS-FLY1 expression in these cells is reduced. So we've now got a chance to say that, well, H&R and PH1 looks like it's required for the expression of EWS-FLY1. We can see that the same occurs at the protein level in that we lose EWS-FLY1 protein. Again, I want to stress in terms of this particular presentation, we're going through and we're doing this with 3 different siRNAs to confirm the specificity of this effect. Now, H&R and PH1 is an RNA binding protein, and that allowed us to come up with a hypothesis that this is actually required for the processing of EWS-FLY1 pre-mRNAs. In a subset of EWS-FLY1 expressing sarcomas, we now know, because if they don't process this correctly, they end up with a premature stop codon. And this is related to the fact that we know, and goes back where we've been able to integrate our RNAi results to all of the data that's now available looking at the transcriptome of Ewing sarcoma, tumors, and cell lines. And what we know from looking at the transcriptome of again, tumors and Ewing sarcoma cell lines is that there are a subset of these tumors that have a breakpoint that means that exon 8 from these cells must be excised to make the in-frame fusion. And so what we discovered by QRT-PCR, shown as analysis here, is that H&R and PH1 is required for this processing event to remove this exon 8 here, but it's the other cell lines that have a different breakpoint do not need H&R and PH1. And this is shown here at a protein level, where if we look at these cells that have this breakpoint that includes exon 8 at a genomic level, if we take away H&R and PH1, they no longer make EWS-FLY1 protein. But the other cell lines that have this breakpoint here or here, they have no dependency on H&R and PH1. Further down from this, we can then look to see, I told you that EWS-FLY1 is a transcription factor, and we can see using this that we can change the mRNA expression of target genes of EWS-FLY1 when we silence H&R and PH1 in one of these cell lines. Again, we're using multiple siRNAs. I'm showing you this to really illustrate how we can use siRNAs now going from a genome level now down to looking at an individual gene and seeing what the effect is on genes downstream of EWS-FLY1. And in fact, EWS-FLY1 can actually repress the expression of some genes, and we see that here, that when we take away H&R and PH1, that actually allows the activation of some of these genes. Now, there's an alternative strategy that we could have used. It would have taken us a lot longer, I think, which was to say, well, what is just actually the effects on a very broad endpoint, which is to look at cell viability. And this is what a lot of people have done. I'm just showing you here is a segue then into some of the studies that have been done like that, which is where actually if we had enough cell lines and we'd screened with H&R and PH1, and what we're measuring here is cell viability, 6 different siRNAs, and we looked at 2 different cell lines. We have the TC32 cell lines, which is the one that we've done most of the studies in, that we know are dependent on H&R and PH1 expression. They are sensitive to the loss of H&R and PH1, but the other cell lines are not. So if we sort of could have used this as a way to get to this information, it would have been, I think, a lot harder, because the only way you can actually get to this information is to screen hundreds and hundreds of cell lines. But that's actually what some groups have done, and so I'm showing you that because if you're working on a particular tumor type of interest to you, you may want to go into these different data sets. Both of these were published earlier this year in July. And so this is a group, the group from Harvard, where they screened, well, I'm going to get this right, they screened, I think in this case, 500 cancer cell lines, looking for specific effects on their viability. So this was an shRNA screen. This was also another shRNA screen. In this case, this is Project Drive out of Novartis, and they screened 8,000 genes in 4,000 cell lines. So one of the concepts is you can take these really, really big data sets and then integrate these with other data sets to find out the types of information I just described to you. But these are very noisy and can be really difficult to interpret. So I want you to be aware of them. Please go look at them in your particular cancer type. You may have interest to you, but just be wary of some of the information. It is quite noisy. Now, because we've spent the last decade using RNAi screens on a very large scale, of course, it was really quite quick that people turned to doing CRISPR-based screens. Again, here using cell viability as an endpoint. And there were two studies that came out last year, both published in Cancer Discovery, where in this case they took, this study took 33 cancer cell lines, looked at basically the whole genome, looking at 19,000 genes, looking at 6 guides per gene, and they were doing these screens where they used about 500 guides per cell. Then the other study took just 5 cell lines and just under 3,000 genes, but they had many more guides per gene. So these were the first sort of really, really large scale screens that were done in multiple cancer cell lines. Now, they got a lot of information out of these that was gene specific, and I'd urge you to go look at these, but there was a problem that came from these that's very relevant to thinking about the off-target effects, in that what they saw was in regions where there was significant amplification, which is obviously something that you see in a lot of different cancers. So in these cell lines where there was a region that was amplified, what you saw then was that any guide RNA that was targeting that amplified region was reading out as a false positive. So what was that? Why was that the case? Well, so here is an analysis here looking at 2 different regions around the MET gene or the YAP gene in either a gastric cell line or in a brain cell line. And this is the data where they looked at the shRNAs, and this is data compared for the CRISPR. So this was from Mons et al. In Cancer Discovery. And what I am showing here is you can see here that this is the shRNAs, and this was really basically a straight line. And really what it was focusing on in these, if you see the orange dots, was around the MET gene, where you really, in this case, the MET gene, or in this case, around where's YAP? There's YAP. This was around the YAP gene. This was working. It should be the green. Where's the green? The green's hidden. I'm sorry. It should be the green. But this had worked exactly the way it was expected to. What was strange on the CRISPR was you saw this line is now no longer focused just around the genes of interest. And what it was discovered was that when the Cas9 is interacting with these amplified regions, you end up setting up a DNA damage response. And this is actually resulting in a false positive response. So it's, again, just being careful that if you're using a cell line for your types of studies using CRISPR and Cas9, you may need to be very aware that if you're targeting an amplified region, this may not be the best strategy for you. Okay, so in the last few minutes, I'm now going to just move to another way that we've been applying RNAi screening, and I've been bringing this in really because of the translational focus of this particular course, which is where we've been using our RNAi, and a lot of others are now moving on into using this in different contexts to look at how to understand drug mechanism of action or combining knockdowns to find ways of chemosensitizing different drugs. And I'm going to talk about one study that we've done in the context of breast cancer, but we've published many over the last few years in this context. So what is the rationale for doing this? And I've obviously done this for RNAi-based, but it would be the same in whatever the approach you're using. What's the RNAi-based analysis for drug activity? Why are we doing this? Well, one of the reasons is for looking at identifying synergistic molecular targets that can exploit complementary vulnerabilities. So you want to, in a lot of cases, increasingly, you want to be able to do combination studies. Those are very, very expensive. So almost no drug that's coming into the cancer field today is going to be used alone. It needs to be done in combination. But those are expensive to do. If we can find out off the bat what would be the right combinations to do, that would be more efficient. And so that's one of the things that we're trying to do. It should allow you to use some drugs at lower concentrations, which will be highly effective. You have the potential to overcome drug resistance, and you can actually broaden the clinical application to many other cancer types. So I'm going to just talk about one of the studies that we published now a couple of years ago to illustrate some of the points that I've raised and how we've gone about doing these studies. So I'm going to talk about TRAIL, which is the tumor necrosis factor-related apoptosis-inducing ligand, which is in part of the apoptosis extrinsic pathway. And what we did, and this was work done with Stan Lipovitz in the women's malignancy branch here at CCR, and what he was interested in looking is finding predictive biomarkers for the responsive trial, for a TRAIL, because they wanted to do trials with TRAIL. And they were interesting also whether we could predict what would be the right combination strategies for using TRAIL. So TRAILs become a very interesting potential target, particularly in breast cancer. So we devised for this that what we wanted to look at was different stages in the way that TRAIL is triggering apoptosis and conduct a whole series of different screens that would allow it to assay what was occurring if we manipulated different genes to alter this response, looking at different points in the pathway, looking at the activation of caspase-8, one of the first effector molecules, then looking at activated caspase-3-7, and then looking eventually at loss of viability. So what this involved was doing was a whole series of parallel RNAi screens. We focused on the kinome and phosphatome plus about another 350 additional genes. And we were going to do these in the context of the absence and the presence of TRAIL. So we're adding TRAIL and seeing what the responses are. This was all done initially in the screening format in MB231 cells. And so we had 3 different endpoints. So one of the absolute critical things, if you're going to look at in the context of a screen, whether it was the screen I discussed earlier or in this one, is you have to have very robust controls to be able to analyze your screening data. And for this, what we had to do was actually obviously was find controls that would work in all 3 assays. In the end, what we were looking for was one that would actually suppress our signal and ones that would activate our signal. So if we can actually use siRNAs to caspase-8 here, this will actually block this response. It's not working. Okay. And then if you use siRNAs to flip, this will actually activate the caspase-8 response. So these were the controls here. They're actually much more robust in the caspase-3-7 assay here. But you actually also have to remember that in the we see you end up with the different set of controls for the cell viability assay. Caspase-8 should have no effect. Let me get this right. That's where we're going to actually block the response in the context of cell viability, and it will be enhanced in the context of FLEP. Okay. So again, what I talked about when I showed you these big graphs, when you're doing the scale, this is again 2 years' work in 6 graphs this time rather than 1. But what we were trying to get here was actually then show the interaction between these different the data sets that we generated. The key thing first is that in the absence of TRAIL, we really see no noise. And this was really important. Your baseline needs to be really nothing. And that was really good. We saw no. Then once we add TRAIL, we see a very broad range of responses. But what we were looking for was seeing where was the correlation between responses between the different assays. In this case, where we're looking at caspase-3 versus caspase-8. Here, caspase-8 versus cell viability. And here, where we saw really nice correlations actually between caspase-3-7 and cell viability. Now, these may not look the best R values in the world, but in the context of very large screening, these are actually pretty good for what we're trying to get out of this. What we can then do is drill down into this data, and there are multiple ways of drilling down into this data. So I'm just going to show you one of the ways that we're able to do this, which is particularly to use pathway analysis or network analysis tools to see where there's a lot of noise here. but can we pull out the signal from this noise? And so what we were able to do here was sort of show where there were nodes, where we saw hits, where there were 3 or more sRNAs that were associated with the phenotypes. We sort of saw nodes around SARC. We saw nodes around BCL2L1, which should be expected, and we were really pleased to see that. We also saw some nodes around FGFR4, but yes, we saw, oh, that was because that was a link to SARC. So we saw these links, and we were able to then go back and start drilling down into these, looking at these in the individual assays. Importantly, we were able to take some of the lead hits we had from this and then now go back and do a sort of a secondary screen, but now increasing the different to number of cells types, including those that are now quite trail resistant. And we're able to now take a cell that's actually quite trail resistant. If we silence with an sRNA, we can now trigger in this case, so I think a change in cell viability. Caspase 3-7 activation. We now see activation in a trail-resistant cell line. So you can actually then take this now into a drug setting. So there are BCL2L1. This is actually a BCLXL, BCL2, BCL1 W compound inhibitor. And now we can take that. So we've now gone from the gene to to a compound, to a drug, and shown that this actually, in combination with trail, is actually able to sensitize cells. So you have a synergistic interaction that was predicted by our RNAi screen. So from our study, which will allow me to segue to something else, is that we actually found about 150 genes that were identified as putative negative regulators of trail-induced apoptosis. And we were able to use small compounds to actually confirm this. Both compounds to SARC and BCL2L1 came from this. But actually understanding this became really complicated. And what we realized is that actually we needed more data from this. And I mentioned earlier about the Translational RNAi screening facility, and I'm going to just mention that at the very end of this. So this was one of the projects that was actually expanded to go into this. So now over 10 years ago, I helped found, and until earlier this year, I oversaw at the 60,000 feet level type of thing, this Translational RNAi screening facility. This basically allows us to interrogate the genome in the same way as people have been doing drug screens for the last 20 or 30 years. It's a fully automated system where we can look at a very range of different endpoints, simple phenotypes, some of the ones I've described here. We can look at pathways using reporter assays. Now, increasingly, we can do high content imaging. That's one of the things that we're working towards, is being able to do that in the lab. So this is actually available to the entire NIH intramural program, but be careful because your PIs need to put in major work to make these, to be able to access this. These are very, very expensive screens, and so there's a full review process for it. But every year we request projects for this facility. To date, they've looked at lots of different endpoints in the context of cancer. I just described one of these, which was the Ewing sarcoma one. They've, as I say, expanded to some of the other studies that started in my lab and gone on to this, and these are all the cancer targets. But they've also done a lot in infectious diseases. You're about to hear about HIV, so I wanted to plug it in there. We've even done an Ebola-based RNAi screen. That was interesting. And then we've looked at lots of different disease phenotypes. So I'm just going to finish there, and I think we've got time for just a couple of questions.
With talents, they normally introduce into the cells plasmid-based or purified proteins?
I think it's both, but immunity purified proteins.
When you have that 21,000 genes and then you design 3 sRNA, so when you use that into your function screen, how many cells do you prepare for the initial?
Yeah. What's the scale up?
Yeah.
So it's all done in 384 well plates. So it will vary from cell type to cell type, how many cells you're going to need from that. So typically, we'll usually, if it's an epithelial cell line, it's about 500 cells per well. Okay. So then you're going to scale that up to hundreds of plates, okay? It's usually done, so I always joke, actually, the screen is the simplest part of it. It's usually done over about a 2-week period. So it takes us usually about a year to get to the point where you can do a screen. The screen is it's 2 weeks work, and then it's another 3 years trying to figure out what it told you, if it's done well. So I'd have to do the math on that. It's been a long time since I actually had to worry about doing the screen, so I can't give it. But scale up accordingly. But it's all done in 384 well plates. Sometimes you've done a tour to NCATs in the past, haven't you, with this group? You've done a tour to NCATs with this group in the past, right? Maybe. Yes. So I don't know if you'll get a chance to see it. Okay. All right. Okay. Thank you.
Well, I'd like to wish everyone a happy Thanksgiving. And our last speaker is Frank Maldarelli. And he's an investigator in the host virus interaction branch, HIV Dynamics and Replications program. He got his Ph.D. from City University of New York, an M.D. from Mount Sinai School of Medicine. He did his residency in internal medicine at the Columbia Presbyterian Hospital, New York. And he joined NIAID initially as a staff fellow, and subsequently he joined the HIV drug resistance program. I'm sure you'll enjoy his lecture on retroviruses. Frank.
Thanks a lot, Terry. It's nice to see you all. I don't think I have anything as extensive as 21,000 different screens or drug screens, but I do have a few drugs, and I'd like to talk about how we got to there. And also, although it may seem a little unusual to have a lecture on retroviruses, or on HIV, I should say, in NCI, I think you'll see that retroviruses probably have a very deep history in our institute. And so I'm very grateful for the opportunity and to Terry to be able to talk about that this afternoon. So what I'd like to discuss really is the molecular biology and replication of the virus, retroviruses in human populations, how they emerged and they spread, how we treat them, and some lessons. And I would just encourage you to interrupt me at any point, especially in case I go ranting in one direction or another. So first of all, to start out with one particular retrovirus, HIV, and this is HIV-1, to give you a sense of the problem. About 36 to 37 million people are infected with HIV worldwide, and I think what you can see is that the majority of them are here in sub-Saharan Africa. In fact, 2/3 of the problem, but also places like Southeast Asia and the Caribbean have a significant disease burden. And finally, there are areas where the disease is expanding, and I would add Central Europe, where it hasn't exploded, but it's certainly increased rather quickly and remained endemic. And I think the thing I wanted to point out is that depending upon where you look, the risk for HIV is different in those places. So for instance, in Central Asia, former Soviet Union, the epidemic was initiated through IV drug use and perpetuated that way, with some commercial sex work, that's CSW, men who have sex with men, hardly any, and then this other group, which I'll come back to. In Southeast Asia, it's largely driven, or at least initiated and propagated through commercial sex work, with some intravenous drug use and some men who have sex with men. And then here in the Caribbean, now the Western Hemisphere, a significant epidemic. But as you can see, not as much commercial sex work or IV drug use, but reported MSM. And in each case, this transmission risk category known as other is either more or less predominant. And I think the reasons for that is that this is really proportional to the stigma of HIV plus the stigma of of being male who has sex with men. So you'll notice that this group other here probably includes all of these individuals who can't disclose their status. Here in Southeast Asia, where things are a little bit more, I guess, freer, where you can say you're a commercial sex worker, but still other 24%, and here in Latin America, 38%. So I guess my point is I'm not so sure what all the driving risks are for HIV infection in today's world because we can't get a handle on all of the demographics. But it certainly suggests that that there's a substantial problem. To understand, what to do about it, I think we need to know a little bit more about the molecular biology and replication. And so that's what I want to talk about next. And I'm sort of curious, like, you guys are all in the NCI? And who's NCI? Who's non-NCI? Who doesn't have hands? All right, never mind. Are you working in labs or are you working in offices? Labs. So pretty much our offices. Any familiarity with things like cDNA? Does that word mean anything to anybody? Okay. It helps. So if we were going to define retroviruses, it's not viruses that came from the 1970s with plaid and polyester. But instead, they're a group of RNA, but these are the jokes. It's Monday before Thanksgiving. I'm sorry, Terry. This is the best I'm going to be able to do. They're a group of RNA viruses that replicate via DNA intermediate using reverse transcriptase. And so they're developed in the they're really identified in the mid to late 1960s. And if you think about it, that's about 15 years, 16 years after the structure of DNA was identified, and maybe about 20 after it was relatively clear that DNA was the genetic material. And in those first 20 years, I think one of the predominant paradigms was that you start with DNA, you made RNA, and then you made protein. And along comes a virus like retroviruses, or what would become retroviruses, what were known as RNA tumor viruses before that. And there, you have a DNA molecule that's integrated into the host, right? It produces RNA in the infected cell. It puts RNA in the virion, and then that RNA in the virion gets transcribed into a DNA molecule before it's integrated in the host. In other words, this is going against the current of the time in the 1960s. And for those reasons, not DNA to RNA to protein, but RNA to DNA to RNA to protein, it was named as a retrovirus because it was going in the opposite direction. So it's not only a different paradigm for replication, but it, as it turns out, it has been, or at least the theory is, it's been quite a benefit. Not just for you molecular biologists who could raise your hands when they heard of cDNA, because the only way to make that is with reverse transcriptase, but perhaps for the other organisms on the planet who don't work in molecular biology labs. And so if you look at all of the current phyla, every last one of them has in its host genomic DNA a copy of reverse transcriptase. And as you know, because I see these folks every year, thanks to Terry, I know how smart you guys are. And I know that you already know that life on Earth probably did not start with DNA as its genetic material, but as RNA as its genetic material. Is that a surprise to any of you? It was what they call an RNA world. The earliest enzymes are RNA enzymes. And so it has been thought for a number of years now that life on Earth started out as an RNA molecule. And as you can imagine, just like us, the first organisms grew, developed, divided, and created, poop, side effects, things that it made in the process of replicating. Those things, in the case of RNA molecules, was molecular oxygen. And as you know, if you've made cDNA, that molecular oxygen is the enemy of RNA. And so imagine living in a planet, this planet, where the only organisms are making molecular oxygen. There isn't a lot at the time, but the more of it accumulates, the more vulnerable your genetic material is. And so the theory, and it's a theory, I'm almost old enough to remember, and Terry is not, what it was like 15 billion years ago. But the theory is that things started out as RNA, and the organisms that were able to persist were those that could convert their RNA genome into something a little bit more stable in the presence of molecular oxygen, and that was a DNA molecule. And so getting from that RNA to DNA, well, you know, they all got a reverse transcriptase. Maybe that's basically a remnant of the process. Jedi Knights, as somebody called them, who saved life on Earth by making sure that the organisms could convert RNA into DNA. So if you ever get out of the NIH, this is a lovely park, Seneca Creek Park up on Clopper Road. Anywhere you look, in the plants or any of the phyla, you'll find a reverse transcriptase. They're all around us. And so if we look at the families now of viruses that use reverse transcriptase, they're divided up into this 7 in the form of this phylogenetic tree. I like to show this around this time of year because some of you may be going to, I don't know, a family event on Thursday, and there'll be, you know, you can look around the table and maybe draw a family tree. Well, this is the family tree for retroviruses. And as you can see, there are about 7 of them. The point I'd like to make is that they're in all kinds of organisms. And the ones that infect humans are down here, one called HTLV for human T-cell leukemia virus, discovered in part here in the NCI. And the other set is lentiviruses. And you can see there are a couple of variants, and one that is probably very familiar to all of you, HIV-1. And then there's a bunch of others here, and these spumaviruses, which don't typically infect humans, can, but don't cause any disease. They're typically in primates. And there's a bunch of other ones. Here's mice. Here's one that is in monkeys. This is mouse memory tumor virus. Here's ones in chickens. And the closest relative to HTLV is actually bovine leukemia virus, is an infection of cows. And so the point I'm really trying to make here is that the phylogeny of the virus doesn't recapitulate the phylogeny of the host. So if it did, then we'd expect that HTLV would be up here with the lentiviruses. They all infect humans, but they are very different from each other. And so just because you infect the same host doesn't mean you are closely related to each other. If we cone down now just on the HIV viruses, or the human immunodeficiency virus, we see that a second time. So here's just all of the viruses that are closely related to HIV-1. And those of you who know of family trees or are going to create one on Thursday, you know that the closest relatives are the ones that have the least horizontal distance. In a phylogenetic tree, the vertical is just to splay all the organisms out, and the horizontal is really here. So to get from HIV-1 to this virus, you only have to go here, down doesn't count, to here. So these 2 horizontal lines is essentially the genetic distance between HIV-1 and this other thing. The other thing is actually a virus that infects chimpanzees. So the closest relative to HIV-1 is a chimp virus. And as you can see, there's another HIV on this one, HIV-2, a totally distinct virus. But look at the genetic distance to get there. There's this, forget about the vertical, and then here and here. But this virus has a very close relative also in the non-human primate community, and it's in a sooty mangabe. So this is a chimp, this is a sooty mangabe, that's a monkey. This virus is 98% identical to this virus. So they're very closely related. This probably around 70%. So once again, just because you infect the same host doesn't mean you're that closely related. This kind of analysis gives us some real insight as to where the viruses originally came from and a little bit how they got there. The other viruses in this group infect, again, monkeys. This is mandrill. And then you get a whole wing of the family that infects. These are goats, horses, cows, and cats. Feline immunodeficiency virus. Some of you may have had some experience with that with pets. This, for reference only, is, and the reference is how concrete are retrovirologists. I mean, you know, you live in your own worlds. I don't know if you work with retroviruses at all or ever met any of the people who work with them, but they're extraordinarily concrete people. They see something, they say something, they name something. And so in the virus, the virion RNA has a specific structure. It has this stuff, something called R, something called U5, a bunch of genes here that I'll come back to, and then at the end, something called U3 and R. And the curious thing is, all right, so, you know, you can have the promoter for anything come afterwards, right? It's possible to have promoter sequences before +1 of the R. This is a little unusual. Having the poly A signal for the RNA come before the RNA, now that's distinctly unusual. But this is the structure of the RNA that gets incorporated into virions. You'll notice another point here. This R on this side and this R on that side, same color, means repeat. In other words, this sequence here of about 150 nucleotides or so is directly repeated on this end. Over here, this U5, this means the unique region in the 5' domain. That's why it's called U5. All right, so given that nomenclature, U3 must be the unique region in the 3' domain. So basically calling them as they saw them, they made this map of the ends of the virus. And then through just standard old sequencing and looking for open reading frames, they could find these genes that confer coding sequences. By way of convention, the genes are always in lowercase and italics. The products, gene products, are always capitalized and in Roman letters. Now that's what's in the virion. Have a look at what's incorporated into the cell. And here you find, and the only reason I do this is because it gives us some real insight exactly how this thing might have gone from RNA into DNA. So if this is the RNA, let's look at it. If we sequenced it out of the host genome, we'd find this same part, R, U5, genes, U3 and R. But now we have an extra piece tagged on. The U3 that was over here is now over here. The U5 that was over here is now also repeated over here. This tripartite element is called the long terminal repeat. This little guy is a terminal repeat. Here's the long terminal repeat. And now what you've noticed that by bringing this over here, we've put the poly A signal at the end of the RNA. And by having the U3 up here, we put the promoter sequence upstream of +1, which is right at the end of R here. So now this looks a little bit more like a conventional eukaryotic gene cistronic organization. Getting from this to this is the job of reverse transcriptase. And so here, just for, again, for reference purposes, is what all those words, what all the genes mean, where they came from, etc. The only piece, I think, really to the top three really are of some use. Envelope, because it's on the surface, it's an envelope protein. Pol, it's the polymerase encodes a reverse transcriptase, a protease, and the integrase genes, all of the enzymes. And GAG, named way before these things were named as retroviruses, as an immunological reflection of telling the viruses apart. It was a group antigen. So it's not to try and make you feel sick, especially this close to a holiday. but it's meant to be an abbreviation for group antigen. Here's how the virion looks in an artist's depiction. It's an enveloped virus, obtains its envelope as it buds out of the plasma membrane. On the surface are surface molecules encoded by HIV, which will be used for interacting with the next host cell. They're complex to a fusion transmembrane protein. On the inside of this envelope protein is a core that includes two identical sequences of RNA. So these RNA molecules, these two RNA molecules are not complementary to each other. They're two 5' to 3' stuck together, and you'll see why. They're both plus-stranded RNAs. And so if we just go through quickly the replication paradigm of the virus, first things first, you have to attach and enter. And I think when this problem was first really investigated, people felt that, well, maybe all it's got to do is interact with the surface of a host cell, and everything will follow directly. And as it turns out, that's a multi-step interaction. So first of all, yes, the virus has a surface protein called GP120 that has to interact with a host cell factor, and that's CD4. It's probably something that you're fairly familiar with, but that's not good enough. It also has to interact with a co-receptor, and in general, the co-receptors are two surface molecules that are common on lymphoid cells, either CXCR4 or CCR5, and that interaction tethers the virus to the host cell, and that is not good enough to get inside. So if all you ever had was that interaction between GP120, CD4, and either R5 or X4, all you would be able to do would be to stick on the surface of the cell. To get inside actually takes a lot of energy, and that energy has to overcome essentially an energy of activation, and that's going to be driven by this envelope glycoprotein GP41. Okay, so fine. If you're going to give a test, this is awesome. I have two distinct steps I can test on. This is just great. Actually, what it's useful for is not just to say, oh, it's a multi-step process. Every time you can find an interaction between a viral factor and a host factor, that identifies a site to get a drug to inhibit it. So in this case, knowing that this protein, GP120, interacts with CD4, okay, well, what if I had a small molecule that could break up that interaction? That's an essential step. Okay, great. And even failing that, I could come back and get another small molecule that perhaps interrupts the interaction between the same molecule, GP120, and either R5 or X4. And then finally, if I screw that up, how about something that inhibits the actual entry, the fusion of the membranes, driving the virion into the cell? So, just this one step of getting into the cell can be broken up into three different steps, each one of which is amenable to a drug or some kind of interruption. And so here's the first of them, that's co-receptor binding. Now this is the envelope binding to R5, and there's a small molecule that's been developed to inhibit that. It's actually available in the clinic now as an FDA-approved drug called Maraviroc. And what Maraviroc does is basically block the binding between the CD4 molecule and GP120. And I know it looks so totally obvious from this, but here's the Maraviroc drug structure. And around this are the numbers of the amino acids in GP120 that it binds to. So that's defining a pocket that that drug interacts with, and once bound, it will now not interact with CD4. I'm moving on to fusion because I don't have a good drug for the CD4 HIV GP120 interaction. And that may seem curious to you because I think if you know anything about HIV, the first thing that comes to mind is CD4 is the receptor. It's been known for close to 30 years. All right, 30 years, you figured they'd be able to figure that out, but it seems to be a difficult process because all of the drugs that have been developed tend to be very ionic and hard to formulate for pharmacologic purposes. Okay, so let's skip that for a couple of minutes. We'll come back to it when we're desperate. But here, if we look at the fusion event, as I say, you have to overcome an energy of activation. So the mere binding doesn't get you in, but the mere binding reorganizes the structure of this protein that's non-covalently bound to GP120 called GP41. And once reorganized, it creates six alpha helices, which act essentially as potential energy built into those alpha helices, which on connecting will cause this fusion event, literally driving one membrane into the other in a series of steps that is well worked out here by this question mark. I don't think anybody really understands one step to another. They know that once GP41 reorganizes, there's a 6-helix bundle. After that, it fuses somehow, but the end result is that now the virion contents become the cell's contents. That structure, let's see if I have that. I guess not. That structure for this 6-helix bundles, you can imagine that an alpha helix, especially 6 of them, might be a druggable target. And as it turns out, all you have to do is reverse the direction of the alpha helix. It binds into these alpha helices and prevents them from transmitting the potential energy in those helices into a kinetic energy driving those two membranes together. It's the drug called T20 is 36 amino acids long. I say that because it still is the longest peptide. It has to be made synthetically. It's the longest commercially available peptide used in for therapy in any disease. So we all know about insulin, but the insulin's isolated and purified. This drug T20 and fuvertide is actually synthesized chemically. So it was quite it's kind of a tour de force to get it done. But you can imagine that if somebody needs 36 amino acids synthesized in an FDA-approved way, you must be a little bit desperate to get drugs to inhibit HIV. And that's actually the case. We'll see when enfuvertide came out, it was sort of at a dell in the development of drugs. So, okay, once we get inside, what happens next? Here's a step called uncoding. Have you ever heard of this step before? Binding, good. Reverse transcription, you probably heard of that. That sounds like an enzyme. What does uncoding sound like? Uncoding sounds like, we're giving this lecture and it's cold outside and you got to take your... It's a very poorly described step in HIV infection. And if it wasn't so critical, absolutely essential, and as yet undruggable, if it wasn't so important, I wouldn't have even mentioned it. But it's the steps in which this core, which was inside the virion, starts to come apart and reverse transcription takes place. Now, why is it so important? And that's here. Once again, it requires interactions between viral and cellular factors. There's a protein called GAG, which makes up this, the core. There's about 3,000 molecules of that group antigen that surrounds the nucleic acid. And the first thing it does in some cells is interact with a host innate immune molecule called TRIM5 alpha. TRIM5 alpha, in the right circumstances, binds to those gag molecules, perhaps as a surface and not as an individual molecule, and prevents the uncoating process from occurring. In an unfavorable circumstance, the TRIM5 alpha no longer can recognize that gag surface, and the uncoating takes place and the virus replicates. And here's why I wanted to mention it, because it is so critical to HIV replication. This comes under, you know, for the want of a nail, or generals are always fighting the last war. If you look at 3 different species, humans, chimps, monkeys, each one of them has their own TRIM5 alpha surveying the inside of the cytoplasm of its infected cells, looking for viruses that are going to come in, not just retroviruses, but other viruses as well, recognize the surface and prevent that virus from replicating, either in the cytoplasm or anywhere else. Okay, so that's nice. Here's, here are 3 different viruses. First, HIV, and you remember I mentioned that the chimp has a virus. It's called simian immunodeficiency virus that infects chimps. And I mentioned one that infects monkeys, simian immunodeficiency virus. Okay. In human cells, HIV infects just fine. The chimp variant infects just fine. The monkey variant infects just fine, because each one of those cases evades TRIM5 alpha. What's with our TRIM5 alpha that stinks so much? I'll explain in a moment. But if you go to the chimp, HIV infects just fine. They go back and forth. That's how we got it. The chimp virus infects chimp cells, no problem. The monkey virus can get into chimps, but very, very poorly. If you look at monkey cells, HIV cannot infect those monkey cells. It has a TRIM5 alpha variant that recognizes the HIV core as it comes into the cytoplasm, prevents that uncoating, and that virus infection is completely inhibited. The chimp virus a little bit, and obviously the monkey virus can infect monkey cells. But, you know, it's kind of for the want of a nail. If we had the monkeys TRIM5 alpha, 37 million people wouldn't be infected with HIV. There are no monkeys that have HIV. There are only people that have their virus. All right, so line them all up. See how many differences there are. It's probably 1 or 2 amino acids. It's just, how could we have screwed this up so badly? And the reason is probably that we didn't screw it up. We are the successes. This room, wherever this is being broadcast today, Dr. Moody, all of us are the successes of previous waves of infections like this one. Retroviral infections that may have decimated populations, and therefore the only ones that survived were the ones that could prevent the infection. Maybe the ones with a variant in TRIM5 alpha. So we, as Charles de Gaulle said, generals are always fighting the last war. We're fighting the last war. We won that one. And with drugs, we will win this one. But the reasons why we have a different TRIM5 alpha are probably because we were selected to have that different TRIM5 alpha. The results of those prior waves of retroviral infections are not just the fantasy of these concrete retrovirologists, but you can find endogenous genes just like retroviruses. They have LTRs, they have GAGs. They're likely the products of previous waves of retroviral infections. So this step that prevents or doesn't prevent uncoating is a critical one for virus replication. And because it's a tough one to drug, people are still working on it, but TRIM5 alpha probably doesn't bind directly to a single gag molecule, but perhaps to a surface. Defining that will define a drug that will make it just as tough to infect human cells as it is for HIV to infect monkey cells. So for the want of 1 or 2 amino acids, you guys could be, you know, doing something else. Once the uncoding takes place, the next step in HIV replication is reverse transcription. And then, so this is a central event in retrovirus replication. This gives it its name. This shows it to be different from all the other organisms. And what it basically does is take these two RNA molecules here in red and convert them into two now DNA molecules, here complementary, here identical. And the gymnastics that it takes to do that are probably not essential. But realize that in making this reverse transcription, what we have to do is start out with a terminal repeat and develop a long terminal repeat, moving our poly-A signal to the 3' end and the promoter to the 5' end. And in doing so, there's a number of jumps and gymnastics that we can go through, but it affects the way you guys make cDNAs. So you know that, you know, sometimes cDNAs come out funny. Here's one of the reasons. Once the reverse transcript is constructed, it has to be integrated into the host genome. The integration process is initiated by an HIV gene product called integrase. See above about concrete retrovirologists. And basically what it does is, first of all, nick the ends of the DNA that it just sequenced so beautifully, take out two nucleotides to make it sticky, cut the host DNA, and integrate it and fill in the last steps of those two nucleotides. Actually, it's going to be 5 nucleotides with HIV to make an integrated provirus using host DNA repair mechanisms. So integrase initiates this process, starts it off, but it needs a DNA repair to actually complete it. Once the host DNA is present in the host genome, the enemy is within, and it will probably be very difficult to inactivate that genome. But I put these both on the same slide because we have wonderful drugs now for both to inhibit reverse transcription and to inhibit integration. So I'll pass through this general evolution, right? So reverse transcriptase, all those molecular gymnastics are quite cool. But let's skip them for a minute and just summarize what RT does. It takes RNA into DNA. It's an RNA-dependent DNA polymerase. To make that second strand of DNA, it has to get rid of that first strand of RNA. So it does that with an RNase H enzymatic activity that's also within the reverse transcriptase molecule. All right, so now it's got a DNA molecule. It has to make the other DNA, right? So it's a DNA-dependent DNA polymerase. An RNA-dependent DNA polymerase, a DNA-dependent DNA polymerase. In other words, it can make DNA out of RNA. It can make DNA out of DNA. The only thing it can't do is make RNA. This is an RNA virus that can't synthesize RNA. This is awesome for exams. It makes mistakes. Oh my gosh, it makes an error. Its error rate of somewhere between 1 and 4 errors per 100,000 bases synthesized. We think, I wish I could make that many mistakes, right? Or so few mistakes. But when the regular DNA polymerases that we humans have are 1 error in 10 to the 9th, 1 in 10 to the 5th, it's fast and furious rates of error rate. So that error rate combined with a replication rate of the virus of about a day, so rapid replication, error prone, translates into a large population of genetically diverse viruses. And you'll see why that's not a product of just a crummy enzyme that's left over from the RNA world. I think of it as a pathogenic determinant. During the course of those molecular gymnastics that we're not going into, recombination can occur across by strand transfers. And so if you've encapsulated different RNA molecules, you can undergo recombination. That certainly occurs in humans. So if we think of the things that we have issues with, right? We have drugs against HIV, but we develop resistance to them. We have an immune system, a much maligned and actually quite effective immune system. In each of those cases, the virus is able to evade both immune surveillance and drug selection, as long as the drug selection isn't so severe. In each of those cases, it's not because the immune system drives a mutation or the drugs cause mutations. In fact, it's the virus that's always been way ahead of us. Because it's rapid and error prone, an infected individual has a very diverse virus population. And so in the presence of an immune selection or a drug selection, all we're really doing is inhibiting the other viruses and letting the resistant ones, the ones that are pre-existing, allow them to replicate. Here's one of my favorite examples of this. And so it's a position in the reverse transcriptase molecule itself. It's at position 151. And typically, there's a glutamine at 151, and it's encoded, as I know you all know, by CAG, right? And now you guys can count as fast, much faster than I can. CAG can go to CTG by an A to T change. CAG can go to AAG by a C to A change. And now I have 3 members in a population using 2 mutations. Well, if these guys undergo 1 more mutation, I can get to an ATG at that position 151. So I've gone from CAG to ATG with 2 mutations. No big deal. All right, so in something that replicates every day and makes a mistake essentially with every virion it synthesizes, the possibilities that these things exist is not bad at all, and the possibility that this might exist as well is not too terrible. All of them are, this is a virus society. This is not necessarily like human society. They're tolerated. Depending upon how frequently they occur, they can be fixed in the population at a very, very low frequency. Okay, fine, Frank, what are you getting at? This variant, this variant, this variant are all sensitive to all of the inhibitors that we have that can inhibit reverse transcriptase. This one is resistant to all of them. So with 2 changes, you can go from a very sensitive virus to a very resistant virus. With one change, you can go from a sensitive to a resistant virus. So in the setting of using a single drug, all you have to do is wait. This is already there. And for single drugs, there's always one of these around to cause mischief. So all you're really doing is selecting for resistant mutations to grow out and inhibiting the growth of the wild-type virus. Integrase has been probably the most recent, I think, enzyme to have an inhibitor, and there's a series of them now. This drug called dolutegravir, a couple of years old now, is probably it may be the best antiretroviral against HIV that we others have developed. And that's because resistance develops, but it probably develops slowly. It's now a part of preferred regimens for therapy of HIV, and it's being rolled out in sub-Saharan Africa in a very big way. So those 20-something million individuals that you saw on that first map, about half of them, I would say about half of them, are going to get a dolutegravir containing regimen. So that very step of integration is quite critical. Once the virus is incorporated and integrated in the host genome, the enemy is within, and everything else that happens, almost every step from that step on, is still a collaboration with the cell. Before, the virus was sort of taking the first step. Here, the cell's taking the next step. So as I said, it doesn't have an enzyme that can make RNA, so the RNA is going to be made by RNA pol 2. cellular enzyme. Once that thing's incorporated, think about it, it's about 10,000 nucleotides. It gets lost. It gets lost in the host genome. Whatever's driving the promoter, whatever factors are available, they're going to bind to the HIV promoter in an activated lymphocyte. So it's just carrying out the program that's already existing in the infected cell. So transcription takes place through RNA pol2, alternative splicing by the splicing machinery, transport by probably CRIM1 RNA transport in some cases and non-CRIM1. Some of this requires the participation of a viral gene called REV. Transcription requires the collaboration with a viral gene called TAC. So in every case, every step of the way, there's always an interaction between the host and the virus. But here the emphasis is clearly on the virus. The RNA is translated just like every other RNA. It gets to the surface of cells using the escort pathway, really co-opting the escort pathway, which brings things into the cells. It reverses it and takes them out. Budding occurs at the cell membrane with the accumulation of RNA, viral proteins, and then one final virus-mediated event when the virus finally buds from the cell membrane. That's not the last event in virus replication. The virus particles now are released from the cells, and they're still not infectious. So the virus is released. That core is actually made up of a polyprotein precursor, and I think this is probably something you're very familiar with, both intracellularly and with other viruses, that proteins are often synthesized as polyprotein precursors and then processed to their final products when they become active. Well, that's true for HIV as well. That final step from going from something that looks like this to something that looks like this requires an HIV gene product called protease. It's a proteolytic enzyme. And it turns an immature particle to a mature particle. These are non-infectious. These are infectious. So in fact, a whole class of drugs for HIV, protease inhibitors that block this step, are literally grabbing victory from the jaws of defeat. Defeat being the cell got infected, okay, fine. It got integrated, okay, fine. It made particles. It's released the particles. They're still not infectious, and we can still inhibit that virus using protease inhibitors. So just because you make numerical particles doesn't mean you make them infectious. So that's the replication cycle. I want to come back to this then to say that there's a host of these HIV-like viruses in animals, and that gives us some real insight as to where this virus came from. Just for a moment, I want to turn to the other retrovirus that's known in human populations, because it is another NCI virus, and this is human T-cell leukemia virus. It comes in a couple of different variants. HTLV1, which can be found sporadically throughout the world, and in areas of high endemicity. And those would be places like the Caribbean and the Far East, especially islands like Okinawa, where the rates of HTLV1 infection have been running as high as 30 to 50%. It's transmitted through breast milk, an entirely different mode of transmission. But because of travel and the use of intravenous drugs, transfusions, it's also set up shop in other human populations. It has a relative called HTLV2, which is endemic in a number of Amerindian and Pygmy tribes throughout the Western Hemisphere and in African tribes. This virus does not cause disease. I have no idea what it's doing there, but it seems to be relatively endemic in a number of populations. HTLV1 is sort of it's the yang to the HIV yin. You already know, because that there are about 36 or 37 million people infected with HIV. Before you came in here, let me just ask you, had you ever heard of HTLV1? If you had, half a hand, two half a hand, one hand. Okay, fine. Now you know it exists. How many people in the world do you think, guess, have HTLV1? And because we're virologists, we're going to think in log terms. 1 people. All right, never mind that. 1,000, 10,000, 100,000, 1,000,000, 100 million, 1 billion. Guess. If 36 million have HIV, how many do you think have HTLV? Terry didn't tell you I'm deaf? Oh, the people in my lab would have told you that I'm deaf. 5 million, a great guess. Come on. I don't tell anybody any answers. Nobody listens to me anyway. Who cares? All right, so let's go 5 million. Who says higher and who says lower? Let's go lower first. Lower, lower. There was half a higher here. Higher? Higher. It's about 20 billion. The numbers of people with HTLV are close to the numbers of people with HIV. What? It's got a lousy publicist? How come is this? And the reason isn't because we don't have so much of it in the United States. It's because if you get HIV, 98, 99% of the time, if you're not on drugs, you're dead. With HTLV, it's about the other way around. Of those 20 million people, only about 5% of them get disease. The rest of them, nothing. They die of something else. The diseases they can get are as a leukemia that's in its name, but also a tropical spastic paraparesis that was first identified in the Caribbean and in South America, but is clearly present in wherever the disease is endemic. So a couple of really prominent diseases, about a half a dozen more rarer forms of manifestations of HTLV1, but you only get 5% of the total population. It's probably been in the human population for hundreds of thousands of years, and perhaps that's one of the reasons why the ratios of infection to disease are so high and so low for HIV. So ATL, the leukemia, has a long latency, about 30 years. About 1 to 2% of all the infected adults. It has a number of, for an ID person, I would call these cool associated illnesses, but they're pretty, they're dramatic and they're debilitating. The therapy for HTLV is being spearheaded actually here at NCI, and there aren't a lot of good regimens just yet. Moving on to HIV, I think we've beaten that to death. If you look at how diverse the virus is in a person, we just said it was pretty diverse. How about in a planet? Well, it's got about 10 different subtypes, clades, monophyletic groups. And the most diverse places, wherever you see something that's really diverse, that's probably where it's been the longest. And that's Central and West Africa. So Beatrice Hahn and her co-workers went back to those areas looking for viruses in animal populations that might be relatives of HIV. And so they knew from zoos that some animals had HIV-like viruses. They went and looked in chimps and sampled chimps. And I didn't know that. I'm sure you guys, you're young, you know everything about endangered and other species. But there are about 4, probably 5 now species of chimps, and they're listed here. And they're geographically distributed. They don't like to cross rivers. So this tends to be a relatively isolated population. Here's another one. Here's another one. And here's another one. And they sampled as many as they could and found viruses that are very, very much like HIV. And their phylogenetic tree is listed here. So here are the viruses that they derived from people. And here's some from Gabon. Here's the CPZ that was from a zoo animal in the United States. Here's from the Cameroon, and then some more from the Cameroon. The point being that our closest relatives to HIV are in chimp viruses. And if you look at the SIVs in monkeys, they're at a greater distance, suggesting that we got this from contact with chimps. And it's not all the chimps. It's the ones that are here and here. And this more distant group doesn't have any HIV. By all accounts, there are no HIVs in that in this group. In other words, these guys probably got away before the virus entered these populations. And so the virus that's in the chimps is somewhat like the virus that's in the monkeys. It's thought that the virus came from the monkeys into the chimps. But before that happened, these guys got away. Well, how did it spread in humans? And this is what's thought to be the cycle. So primates been eating primates for the longest time in Africa, and that includes us eating both chimpanzees and monkeys. Now, you know, it's a pretty common event that poachers will get animals. I don't know how many of you have ever been to Africa and gone to a bushmeat market, but that meat is prepared in a chop house. That stuff goes to a bushmeat market where you can buy, you know, all different forms of primates. Oops. And it's at that market where people will pick up whatever it is, cook it in the home and whatever. But I think from either capturing it, if you don't shoot it, or chopping it up and perhaps preparing it, you can be exposed to blood and body fluids. And it's that exposure that's thought to represent the transmission to humans. If you look at all the different kinds that are in humans now, you can use bioinformatic techniques and Bayesian analyses to trace the origin of these viruses. And it's thought that they entered the human population not in the early 1980s when the epidemic exploded in the United States, but probably much further back, and perhaps in the period that goes back to the colonial period in Africa, the late 1890s and early 1900s. And so the transmission by blood and body fluid, you know, or transfusion or vaccination, iatrogenically spreading the virus, mother to child, it could be spread. But those are the biologic methods. There are non-biologic ones that also probably participated to the transmission of or to the development of the epidemic. So these things will get the virus into the human population. What gets it to be an epidemic? Let's face it. As I said, primates been eating primates for the longest time. How come HIV, how come we got this lucky in the last 100 years or so? And that's probably because of other factors affected the spread of the virus, both political, economic, and perhaps multiple epidemics. So here's a map of Africa in the late 1800s, and you can see it's divided up into, you know, maybe 4 or 5 groups. And then here, after the colonial period, a number of different countries, and the whole thing sort of coalescing around different European countries that have interests there. And then finally, what happened over the course of a very short period, the late 1940s to upwards of the 1970s, when all of these countries really became independent. And this period included a lot of political upheaval, population movement, malnutrition, and malnutrition is, for an ID guy, is immunodeficiency. And so relative immunodeficiency in populations may have contributed to the spread of the virus. This was probably an important factor. Here, across the bottom is years in the 1900s to 2010. Here, the human population in millions in Africa, and you can see a very large bump from below 10 million to over 100 million in less than about 30 or 40 years. And this great increase in population probably contributed, knowing that the spread of HIV in Africa is not because of MSM. There are equal numbers of men and women with this disease. So whatever guy it is, many people also got you exposed to HIV. And then finally, the development, really the industrial development of Africa. This is something called the Trans-Africa Highway. And along this highway, which connects the east to the west of Africa, are places where, you can refuel, there are hotels, there are brothels. How did it get into the United States? It looks like from some very detailed analysis that the virus didn't come directly from Africa, but perhaps made a stop in Haiti. So the viruses in Haiti during the 1980s are older than the virus is in the United States from the same period. So there was a lot of diplomatic interactions between Haiti and Central Africa, and it was thought to come over on those by those means. In the United States, here's an old CDC map. Each of these yellow points represents about 30 cases. This is 1983. There were about 1,000 cases reported of AIDS in the United States, AIDS being the advanced disease. And here, 2 years later, it's gone up a log. So 1,000 to 10,000. In 4 years, another log to 100,000. You can see, like flying over the United States, places of high population. And then by 95, 6 years later, only another half a log. So you can see that big increase, 83, 85, 89, a log. And now here, 6 years later, we only get a half a log. And now in the United States, there are probably only about 1.1 million cases. So the epidemic occurred, the epidemic spread occurred in the 1980s. Today, the transmission is still men who have sex with men, heterosexual now about 30%, and IVDU about 12%. Importantly, the virus is spreading out of those areas of high population growth. 12% of cases are in locations with populations less than 50,000. Women now comprise 25% of all AIDS cases, and the epidemic is aging. How about therapy in the last 2 or 3 minutes? This was a billboard on the side of a bus when Terry was in high school in the 1980s. And so this was designed to try to get people tested. And the point was, if you get the AIDS virus now, you and your license could expire at about the same time. There was no therapy in in those middle 80 periods. All right, over the course of, I guess what, to my mind, looking at all of you, looks like your lifetime or less, a number of drugs, probably 28 or 29 independent antiretrovirals have been developed. And for the purposes of the slide, they've been divided up into classes, things that inhibit reverse transcriptase, protease inhibitors, integrase inhibitors, those co-receptor and the fusion inhibitors. And you can see that early period dominated by a single drug, AZT, and then DDI and DDC, some pretty poor NRTIs, as well as stabudine. It's not until 96 that we get multiple drugs that people will be able to tolerate at any given time. And so if you think that a single mutation can be selected with a single drug, it's not until you get 3 drugs on board with different selection that select for different mutations that you can get an effective suppression of virus. This is really 1996, 97, 20 years ago, becomes the watershed moment in treating HIV. Theoretically. But what's it look like now? And this is the situation that you have to fix, okay? This is what's known as the Gardner continuum of care. Gardner's a guy who said, let's look at the whole picture at 30,000 feet. We think that maybe there are about 1.2 million people in the United States with HIV infection. We think that we've identified about 86% of them. All right, so we're missing 14%. All right, 14%, that's our problem. We got to go out and find another 14% of individuals who have HIV. We find them, we can stop them from transmitting. Okay, here's a number, here's a problem. Our solution is better surveillance. All right, so fine. Then our problem is done? No. How many of those people that we have are actually engaged in care, in medical care. It's only 40% of this 1.2 million. So if 14% of undiagnosed people is a problem, here's another one. 46% of those people who are identified are not engaged in care. How many of these are prescribed antiretroviral therapy? This is now up to 40%, but still, we're missing 46%. And of those people who actually get the therapy, how many of them are virally suppressed? A third. So if 1.2 million is our problem, about 400,000 people are the current solution. And so at each step of the way, we identify a challenge, a limitation, and an opportunity for you guys to do something really useful. So in the last several years, there have been a couple of key advances. Now, for people who are at high risk, we actually prescribe antiretroviral therapy, 2-drug therapy for prophylaxis to prevent transmission. We've found out that we need continuous therapy to keep all the side effects, not just the HIV associated effects, but all the side effects of the virus to be suppressed. And that the earlier the therapy is initiated, the greater the preservation of therapy. Once again, the faster you identify people and get them on therapy, the more of their immune system you'll preserve. The next advances, I think, will come in vaccines and cures. Just some final lessons. The obvious ones, right? Viruses are bad and should be avoided, except maybe when they save the planet, right? And maybe if it saves you from the next virus. Epidemics are not single events. They evolve. Antivirals are useful. As early as possible, adherence is essential. So I'll stop there and maybe take questions, and sorry for going over.
For the HIV integration studies, would they just integrate randomly, or do they have any?
Terry, I just want to say I did not ask him to ask that question. We've been very, very interested in integration sites. And since you asked, and Thanksgiving's not for another 3 days, enjoy. So what we found, and other people have found, is that HIV integrates with some preferences. It really likes open chromatin. It likes transcribed genes, not heterochromatic genes. And other than that, there aren't too many preferences. There are a few areas of the genome that it likes to integrate into, but I wouldn't call these, I would never call them hotspots for infection. Steve Hughes in the NCI has made a life of studying this kind of thing. And he has some I'm very concrete, but what I think of as astounding new results, looking in cell systems, humans, and primates, and finding that HIV will integrate pretty much in the same genes, regardless of the circumstances. So whether it's my patients that he's studying, cell lines that have been infected with HIV, PBMCs from monkeys that have been infected with HIV, the monkeys themselves, even though the genes are on different chromosomes, these chunks of genes may be translocated to anywhere in a cell line. They still seem to have the same rates of integration. And so these genes, because they're expressed to the same degree, I think that may be one of the reasons. There's an extraordinary interest in looking at the structure of the human genome in the interphase nucleus. How all of those chromosomes, once they're open, are they near other chromosomes? And I think some people feel very strongly that some of these integration events are spatially located within the nucleus. And that, I think, is going to be a very interesting thing. For a couple of integration sites, and I would say 2 that we found and I've studied, and maybe one more, they seem to have, integration seems to have a driving effect on the persistence and expansion of those cells.
With that, we shall close.
Okay.
Thank you, Frank.
All right, thanks a lot.